I will give you an application overview of tissue cytometry using the TissueGnostics platform by TissueGnostics with a focus on immunophenotyping and quantifying the tumor microenvironment in situ. Just a few words about TissueGnostics. Who are we? In fact, I have originally been trained as a cell biologist and worked in prostate cancer research. And the development of the prototype technology for TissueGnostics was part of my PhD thesis at the Medical University of Vienna. The company itself was founded in April 2003, so this year we celebrated our 17th anniversary. At the moment, we have installations in 31 countries on all continents except Antarctica. The headquarters is in the city of Vienna in Austria, in this lovely building very close to the city center. But we have branch offices or subsidiaries in Romania, at the US East Coast and West Coast, as well as in China. We are now approaching 1,600 reference publications from our clients and users in 58 countries around the world. And I would like to mention that TissueGnostics is an ISO certified manufacturer of medical devices. So we're not just a software company, we are a medtech company. We have always been very closely linked with academia and academic research. And I'm particularly fond of the fact that TissueGnostics has been industry partner in up to now 5 EU-funded Marie Curie innovative training networks. The first one dealing with digital pathology. This was headed by a group from Spain and University of Nottingham in UK. Then a second one dealing with the calcium sensing receptor as a key molecule in a number of non-communicable diseases like breast cancer, diabetes, asthma, or Alzheimer's. Then this was headed by Medical University Vienna, Alcatraz, which is a link to Susan Turner in Cambridge, because she was the head of this EU project on translational oncology using the anaplastic lymphoma kinase. The number 4 was INSEM, the European Network on Cell Migration Studies, headed by Technical University of Aachen in Germany. And the most recent one is called Helical. This project focuses on big data analysis using machine learning technologies and is headed by a group from Trinity College in Dublin. And in all this project, TissueGnostics contributed or still contributes in the level of tissue cytometry, of image cytometry, both using classical approaches as well as machine and deep learning technologies. What is TissueGnostics? TissueGnostics is not a single instrument. It's a platform that can be configured to the needs. There are configurations available for bright field scanning, fluorescence scanning, combinations thereof, inverted systems where we can also scan well plates in addition to slides. So you can do live cell imaging or time kinetic experiments. For high throughput scanning, there is a version with a slide loader for up to 120 slides. And each fluorescence configuration can be equipped with a spinning disc confocal. So we can also do whole slide imaging in confocal mode. Here we see such an example, which we have received from Janelia Farm Research Campus, one of our clients in the US, belonging to the Howard Hughes Medical Institute. What we see here is a section through an entire mouse brain. And the individual tiny rectangles that you see here are in fact not tiny, that are the individual high power fields of view acquired with a 20x plan apo objective lens. In order to scan the entire mouse section, 350 fields of view had to be acquired. Each field of view consists of 4 color channels, and each color channel, in case the set stack option is chosen, consists of 21 layers, so main focal plane plus 10 layers above, 10 below. That gives a total of 29,400 images and 115 gigabytes of compressed data. Scanning of this entire set stack takes 1.5 hours, but as I said, 29,400 individual images. When we scan only the main focal plane, then it takes 8.5 minutes. In this virtual slide, we can then zoom in and navigate through it, similar to Google Earth. You can zoom in, zoom out, navigate around, and observe the details, the cellular and even subcellular details. In another example from Jacobs University in Bremen in Germany, we also have a full mouse brain section. And when we look on this region more closely, this is the so-called third ventricle, then we see here up to which magnification or which resolution you can actually zoom in. This is the result of scanning using a Plan Apo 63X 1.4 numerical aperture oil objective lens. Now, the main thing that TissueGnostics offers is not scanning. Scanning is a prerequisite, and scanning at high quality is even more important for accurate analysis. But what makes TissueGnostics is the software for doing image cytometry. So I will now guide you through my presentation with different applications, and we start with the quantification of nuclear markers using a simple bright field immunohistochemistry protocol using the peroxidase staining technique. What we see here is a column sample stained for a well-known marker in brown, Ki67, so a proliferation marker, and the counter stain in blue is hematoxylin. This labels all the nuclei, and the obvious question is how many cells are proliferating, or in other words, how many of the blue nuclei are also stained in brown. Well, we made a survey using this sample and asked scientists around the world, more than 200, what they think how many cells are positive in the sample by estimating and giving a percentage value. And believe it or not, the answers that we received ranged from 1 to 40%. So some people think 1% of the cells are positive, others think 40% are positive. And this indicates the main problem that is always associated with visual evaluation. We get a range of estimations rather than a reproducible measurement. With our TissueFacts platform, we use the nucleus as a so-called master channel in order to identify all individual cells. You can see here the yellow contours, what the software has identified as single nucleus, and then quantify by means of image analysis how much brown and how much blue is present. In this case, it's not restricted to brown and blue. You can quantify any color combination and also fluorescence, as we will see in the next example. But the important point is what you get here as a readout looks like this. It looks like a classical flow cytometry scattergram or dot plot, as this is called, where we have here on the x-axis the mean intensity of hematoxylin, and on the y-axis the mean intensity of the brown marker, in this case Ki67. It is not a flow cytometry dot plot, it is a tissue cytometry analysis, and each individual dot that you can see here represents a single cell in that tissue section. The line that we see here is well known in flow cytometry and very important. It is called the cutoff. It separates unspecifically stained cells from our specifically stained or positive population. So what we have here is the Ki67 positive population. And as you can see, these are 14.5%. The cutoff has to be set, like in flow cytometry, based on an isotype matched negative control. This is the scientifically established procedure to set the cutoff. And the important thing is, whenever we do this analysis on Monday morning, on Wednesday noon, on Friday afternoon, in Cambridge, in London, in Vienna, in Brisbane, we will always get the same result, a measurement, rather than a wide range of estimations. This is how we analyze markers that are expressed in the nucleus, like Ki67, is a nuclear marker, by using the nuclear stain as master channel. In fact, we use the same approach when the molecule that we need to quantify is expressed in the cytoplasm. So when we're looking on cellular compartments, we also use the nucleus, but then generate a secondary measure mask around the nucleus. And this will allow us to quantify cytoplasmic markers, as we can see here. This is an example from skin cancer research. We have here a skin biopsy taken from normal skin. The marker stained is called COP1, but that's actually not relevant. Relevant is the methodology of analysis that we do here. After UV irradiation, we can see this marker, the expression of this marker in the epidermis increases in two ways. First, the amount, here we see there's a constitutive expression. After UV, the amount increases. And the second way is that more cells become positive. So normally people look at this and then estimate again and say, this is 1+ positive and this is 2+ positive. Maybe your colleague comes and says, I guess this is 3+ positive. And in the end, you conclude it's 2.5+. So rather than discussing and assigning pluses to your samples, our tissue cytometry platform can give you an observer-independent measurement. Here we see the result for the normal skin biopsy, and on the right-hand side, after UV irradiation. Both scattergrams have the same scaling. On the x-axis, the intensity of the nuclear marker, the DAPI in this case. On the y-axis, up to 150, the staining intensity of this COP1. What we see at a glance is that in normal skin, 16.8% of the cells are positive, while this increases after UV irradiation to 44.7%. What we also see is that the amount, the intensity of the staining increases. Here we have in normal skin a maximum up to 90, and then we see here a shift towards higher values. So what we visually perceive as brighter is reflected by this shift towards higher values. And the inserts here, known from flow cytometry as the quadrant statistic, because the two cutoffs here separate our diagram into 4 quadrants, the upper left, the upper right, the lower left, and the lower right. And this quadrant statistic shows us the mean intensity in each quadrant, the number of events in each quadrant, obviously the relative percentage, but also the density, means cells per square millimeter of tissue analyzed. So this is a normalization to the size of the sample, which makes then also comparison between sections of different size very easy. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells between the virtual slide and data space. So we see here a renal allograft rejection stained for T cells in green and monocytes in red. When we click on a green cell, you see it starts blinking here, green positive, red negative. When we click on a red cell, it's green negative, red positive. And you can do this for every cell in your sample and know exactly where this cell is located in each of the scattergrams. Following our motto from image to data, we consider this a step forward and thus call it the forward connection. It also works the other way around, from data space back to the image. We can create a gate which is a selection of a subpopulation of cells. So this subpopulation is then highlighted here in other scattergrams in a specific color, in this case orange. But we can also visualize here with red contours where the cells in this gate are located in the original virtual slide. So we have a link between a subpopulation, a specific phenotype of cells, and its localization in the tissue. So a link between a phenotype and morphology or histology. And this is one application that is very important in tissue cytometry, that we can determine the immune status in situ. In flow cytometry, we can determine this immune status of a patient systemically in blood. With tissue cytometry, we can do the same thing, but in a specific organ, a specific tissue. This is also referred to as the phenotypic characterization of tissue infiltrating leukocytes. Here we see an example from a client at University of Western Australia in Perth, who have kindly provided this sample, this tissue. It's a 4-color staining. TAPI labels all the nuclei. Then CD4 in yellow are the T helper cells, CD8 cytotoxic T cells, and FOXP3 in green. You may be aware that CD4 FOXP3 double positive cells are referred to as the Tregs, the regulatory T cells. And this is the aim of this study, to quantify this population of T helper cells and identify also their localization in the tissue. So as I said, we use TAPI as a master channel. The green contours here indicate what the software has identified as a single nucleus. Then we expand the mask a little bit and measure around each nucleus, because CD4, CD8 are membrane stainings, membrane-bound stains, while FOXP3 is a nuclear marker. And then we measure for each nucleus how much yellow, how much red, how much green is associated, and can show this in such type of diagrams. Here we have TAPI versus CD4. This is the CD4 positive population. We have here in red all those cells which are CD4 positive. Some are very low, yellow positive, so CD4. They will be somewhere near the cutoff. Others, like this cell, have strong, prominent staining. They will be somewhere up there. So you can read the scattergram really like in a fax plot, in a dot plot from flow cytometry. The further up, the more of signal for this marker is present. The further to the right, the more of the marker shown on the x-axis is present. The same is can be done for CD8, and finally the co-expression of CD4 and FOXP3 is given. So this is the cutoff of CD4, this is the cutoff of FOXP3, and these two cutoffs separate our diagram into these 4 quadrants. Here, upper right are the double positive, lower left are the double negative, upper left and lower right are the single positive. And we see here in percentage how many cells are double positive for CD4 or FOXP3. So what's the percentage of co-expression? On the single cell level, and please keep in mind, each dot represents a single cell in a tissue section. We have now seen that we can quantify markers which are expressed in the nucleus, like Ki67, or in the cytoplasm, like CD4, CD8. FOXP3 is also a nuclear marker. With our TissueFox platform, we can also analyze subcellular compartments or signals, like FISH or RNA scope. But I would like to show you a very interesting application of intracellular parasites. This is an application that has been developed in collaboration with the Institute of Immunology at the University of Regensburg in Germany, Uwe Ritter's lab, and using leishmaniasis. So I will not talk now about this disease or the biology of infection, but about the methodology to quantify them. In short, These are protozoan parasites that are transmitted by mosquitoes. They infect macrophages, and patients suffering from these infections really encounter severe tissue damage. Now, in this study, cultures of macrophages were used, and in Regensburg, they are working on an inverted tissue FACS system, so using microwell plates. We see here one virtual well, not virtual slide, but it's from a well plate, so it's a virtual well from a 24 well plate. The individual rectangles here show how many fields of view had to be acquired. That's 20x in order to cover the entire well. And what we see is the DAPI stain. The big blue blobs are the nuclei of the macrophages. The small blue dots are the nuclei of the parasites. So what has been done? First, the nuclei of the macrophages were identified. These are these inner rings here, these contours. And then a secondary measure mask was created, a ring mask, which we see here, around each nucleus. And in this ring, the nuclei of the parasites were identified, as we can see here. Now, this algorithm is a mathematical procedure. An algorithm does not understand what is a parasite. It just analyzes groups of pixels in the image. Therefore, we need to fine-tune the algorithm and set the sensitivity. The lower panel here shows the control. This is a population of cells, of macrophages, which are not infected. So here we should not find anything. What we see here in the first image in the lower left, these are actually false positive dots. So the algorithm found here just some noise, some background structures in the image. So we adjust the sensitivity of the algorithm in a way that it does not find anything in our control population, and then apply the same setting to the test sample where the parasites, Leishmania major, are present. And what it finds then, these are really the specific parasites. In this study, the automated analysis has also been compared with manual counting, showing a very good correlation. And by double staining, we were even able to distinguish between live parasites and dead parasites. So we could count the cells, we could count the number of parasites per cell, but also determine how many parasites per cell are dead and how many are still alive. I'm sure nobody of us has missed the fact that we live in a pandemic world, and a virus is capturing all of us, more or less. And therefore, it might be of interest for you to know and to see how tissue FACS can also be used in viral research. The first application, this is not coronavirus, but influenza, and very similar to what corona does with the effect of pneumonia, is shown in this publication, where the viral load in individual epithelial cells of lung tissue has been quantified in situ. So this is one application. You can quantify how many cells, like in the parenchyma or in a certain structure, in alveoles or whatever, in glands, whatever tissue you're working with, are infected, and what's the viral load per cell. But at the same time, you can combine this with what we have seen in the previous example, the immune status in situ. You can also quantify the immune response against this in viral infection. The second example is Zika virus. Here, individual virus molecules have been quantified in the individual cells. Dengue virus, we see again quantification of viral infection. But what I want to point out here is when you look on the number of infected cells, 0.06%, 1.39%, and so on. This makes hopefully clear that with this technology, we can not only analyze cell populations which are big, or analyze markers which are present on 50% of all cells. No, we can also quantify minor cell populations, very small numbers of cells, almost like single cell rare cell detection. We're living in the time of personalized medicine, or the age of precision medicine, where it becomes ever more important to stain more and more markers in parallel. So classically, when I was a student, most people were happy when they could do a two-color fluorescence staining. Nowadays, two-color is not enough. We need to go to high numbers. In flow cytometry, they stain 20, 30 markers simultaneously on the same cell population. And tissue cytometry is also developing in this direction. One approach to increase the number of markers is multiplexing. You may be aware that in fluorescence microscopy, a limitation comes from the fact that fluorochromes have emission spectra, which limits the number of fluorochromes that can be applied at the same time. Because when you have too many fluorochromes, the emission spectra start overlapping and a specific detection is no more possible. But what you can do is that you stain with 4 markers in your sample, then remove the stain and stain the same sample again with the same fluorochromes but different antibodies. So each fluorochrome has a different meaning in each staining round. This is called multiplexing. There's a nice overview paper from Yale University, Kim Blenman, from April 2019, published in Cytometry, where this procedure of multiplexing, in particular to quantify or phenotypically characterize cells clustered as immune cells, was presented. And I don't have the time to go through the details, but Kim describes here the whole process of staining, scanning, analyzing, removing the stain, and restaining. And this can be done multiple times, so that in the end you have 10, 15, 20 markers stained on the same cell population. And you end up here with such a diagram, like you see CD4, Ki67, coenzyme B, IL6, FOXP3, and other markers. And all these dots represent single cells in a tissue section, and they are all stained on the same section. So we're not looking at consecutive sections, but at consecutively stained identical sections. This is called multiplexing. Another approach to increase the number of markers on the same sample, so that are stained on the same sample, is multispectral imaging combined with spectral unmixing, where a liquid crystal tunable filter is used in order to acquire a lambda cube. You might be familiar with the term Z-stack from confocal imaging. In a Z-stack, we look at the same region in different focal positions. In a lambda stack or lambda cube, we look at the same region in the same focus position, but at different spectral intervals. Here we see an example before and after unmixing. So on the left-hand side, this is the raw data set, the lambda cube. You see it's oversaturated. There is unspecific staining, or not actually unspecific staining, but spectral overlap. But after unmixing, we get a very nice and clear sample, like here, mouse spleen labeled with 5 markers. This spectral unmixing was used in a recent study that was published just in October this year in Cell, cell has impact factor 38.6, where the immune response against SARS-CoV-2 was quantified using TissueGnostics technology in the lung tissue of COVID-19 patients. And in order to do this, multispectral analyses of 7 markers were applied, which are shown here. And I don't have the time to go into detail. Talking about this paper would alone take an hour. But the take-home message of this study, which comes from a group in Boston, unfortunately is not very good, at least not good in terms of the current pandemic. Many governments around the world currently wait or hope for herd immunity. And in fact, this paper suggests that the SARS-CoV-2 virus escapes the immune system in a way that these follicle with germinal centers are not formed. So formation of herd immunity is impaired. If you want to know more, I recommend you this publication. It's anyhow online and published since a bit more than 1 month. Now, in TissueGnostics, we have always had a focus on all aspects of image cytometry, not only both on the classical, but also on contemporary approaches using machine learning techniques and even deep learning solutions. In the latest TissueGnostics Suite 7, which we launched a few months ago, we have a new algorithm for nuclear detection, which has been developed based deep learning technology. And this focuses in particular on lymphatic tissue, where the cell density is very high, as we can see here. When we look on the DAPI channel only, you see, because it's lymphatic tissue, the cells are densely packed, nucleus touching a nucleus. And this is really a very difficult situation from the image analysis point of view, to identify here single cells accurately. And with this machine learning technique, deep learning solution, we really get impressive results. Machine learning is also used for classifying histological entities, so structures in the tissue, not based on single cells, but on morphology, like we can see here in this skin sample. And in here we have classified 7 types or 7 compartments in the tissue. 7 classes have been defined. Just a few examples need to be highlighted. So by making like a circle or drawing a line and saying this is typical, so this is how my class 1 looks, this is how my class 2 looks, and so on. Then the training is performed and applied to the entire sample. Now, we have seen and heard that with this tissue cytometry platform, we can scan bright field samples, fluorescent samples, confocal, multispectral, multiplexing. Excuse me. We can analyze markers expressed in the nucleus, in the cytoplasm, subcellular structures, multicellular structures. This now, when we combine all this, we can do something that is called contextual image cytometry, also referred to as spatial immunophenotyping. We see here an example of a colorectal cancer stained with 5 markers, dark blue for DAPI, light blue for cytokeratin, labels the tumor area, red again CD4, and green FOXP3, and white Ki67. We identify the whole tissue or the nuclei, the tumor area showing orange. And these measurement masks can then be combined and allow us to address or ask all kinds of questions. The question here was, how many CD4 positive cells are present? How many are in the tumor area? How many are in the tumor stroma? And how many of those CD4 positive cells are also FOXP3 double positive. And we can see in this catagram, which has an input on CD4, that means all dots that we see here represent CD4 positive T lymphocytes, T helper cells. On the x-axis, we see the size of the nucleus of each cell, and on the y-axis, the distance in micrometer. So there is no positive negative distinction here. It's a distance measurement in micrometers, and the distance that is measured is distance from the next tumor area. And we can make here gates like 1 to 25, 25 to 50, 50 to 100 micrometers, and so on. And in this quadrant statistic, you see now in total 7,234 cells, CD4 positive cells were analyzed. 2,267 or 31% are within 25 micrometer of the tumor area. 1,593 or 22% are within 25 and 50 micrometer, and so on. And we see here, the further we go away from the tumor, the lower the infiltration with T helper cells. So these cells, and this is not a guess, it's a measurement. These T helper cells really target the tumor. They're not just somewhere by chance. They are really specifically targeting the tumor. Now we can use the backward gating functionality that I described earlier and visualizing this image in red, now all those CD4 positive T helper cells, which are just outside the tumor and not further away than 25 micrometers. So what we see here in terms of measurements, in terms of numbers, can also be visualized. These are the CD4 positive cells 25 to 50, 50 to 100, 100 to 150 micrometer away. So we can quantify the expression or co-expression of markers on the single cell level throughout the tissue and do this in a spatial context. We know how many cells are there and where are they and how are they related to each other. And this ultimately leads to this application of immunophenotyping or spatial phenotyping of tumor and the tumor microenvironment. We can also display these distance zones here in different color shades, and by this then measure how many and quantify how many cells are inside the tumor and how many are outside in the tumor stroma and how far are they away. And because this can be combined with the phenotypic characterization that we have described earlier, or just seen in the previous slide with CD4, FOXP3, but you can also look on other markers, CD8, CD45, CD68, whatever, then we can distinguish, like here, between cytotoxic T cells, effector T cells, and regulatory T cells. So you can look on the total T cell population, but also on any subpopulation, and really get a measurement of how many cells are in the tumor, how many are in the stroma. And this can be combined with this multispectral imaging. We see here a colon cancer sample. This is the original image. You see there's a lot of spectral overlap. It's not very clear, but after unmixing these markers, we get a very nice and clear and distinct staining pattern for all those cell populations. Here we see the markers, the individual markers as separate channels. And as I said before, this forward connection function, when you double click any cell in your virtual slide, it starts blinking and you see exactly where this cell is located in these catagrams, and the PD1 CD8 double positive cell that we are looking at here. Now, in more detail, here we see the CD68 positive macrophages, right? In this gate, and the backward connection shows them all with a red contour. And we look on the CD8 cytotoxic T lymphocyte, on the CD4 T helper cells, on the CD4 PD-1 double positive T cell population, which is just a few. There are just a few here. And in this slide, we see a population that is first PD-L1 positive. So these are the cells in this gate, visualized by in green color. This gate, the PD-L1 positive cells, are selected as input gate for the second scattergram. So all the dots that we see in the second scattergram come only from this PD-L1 gate. So PD-L1 negative cells are not shown here. In this scattergram, we show now CD4 versus PD-1, which gives us a separation into these 4 quadrants. And here we choose the lower left quadrant, so the CD4 negative, PD-1 double negative population, and select this as input gate in our third scattergram. So here are only those cells which come from the lower left quadrant. And in this, because in this scattergram are only cells which are PD-L1 positive, also the cells here are PD-L1 positive, because this gate depends on this gate and this gate depends on that gate. So what we see here then in the lower left quadrant, this is CD8 versus CD68. This is the double negative population. And when we visualize them here, that means now in red contour in the image, we see those cells which are PD-L1 positive, CD4 negative, PD-1 negative, CD8 negative, and CD68 negative. Of course, you can do this for any combination that you might be interested in, but this shows you can do a very, very detailed quantification on the single cell level and always with context to the histology. So with the spatial information based on analyzing multiple markers stained on the same sample. That's why it's called spatial phenotyping. To conclude my presentation, I would just like to mention that this technology has been evaluated multiple times. The first publication was done by myself already back in 2006 and published in Cytometry. The instrument is called TissueFax, but the methodology that this offers has been called microscopy-based multicolor tissue cytometry, MMTC. And we combine this here with a standard FAX analysis by using peripheral blood leukocytes stained for CD3, CD4, CD8. FAX instrument was a Becton Dickinson FAX scan, and MMTC was our TissueFax. And here, when we plot the population, the results of the population analysis on FAX and on TissueFax against each other, then we get a correlation coefficient of 0.981, so close to identical. Others have also made comparisons, like a group from University of Western Australia, where FOXP3 positive cells have been counted by 3 independent observers. As usual, when 3 experts count, you get 3 different results. A median was formed from these 3 manual counts, and this was compared with the software analysis, overall showing also a very good correlation coefficient. So I hope I could give you an introduction and show that TissueFax cytometry supports us by moving from image to data and thereby helps to increase observer independence and reproducibility. I invite you to have a look at our website, TissueGnostics.com, where you will find a searchable database with all publications, at least those which we are aware of, You can also search for certain markers, certain tissues, certain diseases you might be interested in, and see who has already worked on this using a technology from TissueGnostics. This is also helpful in finding references for your own publication. I would like to thank our collaboration partners from academia, in particular from our European Marie Curie Innovative Training Networks, like Susan Turner from Cambridge, who has kindly invited me to give this seminar today. And with this, I would like to conclude my presentation, and I hope I could show you that with TissueFax you can explore cells, the fundamental unit of life, with a precision that inspires. If you have any questions, I'm happy to discuss now, but you can also contact me always later at my email address, which is shown here, rupert.ecker@tissuegnostics.com. Thank you very much for your attention.
Thank you so much, Rupert. That was really interesting. I can't see any particular questions in the chat, but I see Mario has his hand up. Mario, if you'd like to unmute yourself, or if you're just reacting to the talk.
Okay, I am going to, I don't need the video, but yeah, I am going to. Hi, thank you so much, Rupert, for the talk. Really interesting work. I would like to ask you, there is some data available, some public data where to explore this kind of this kind of analysis with like repositories like TCGA or something similar.
Sorry, can you repeat the question? If there are public data available?
Yes, yeah, it will. For example, these papers you have shown the data is public or is not possible. It's possible to have access to some kind of this type of cytometry data. For example, in the you have shown a study of colon cancer. Will it be possible to have access to this type of data? There is public data sets of colon cancer with this.
The image data set.
Yes.
Yes. Well, most of the data that I have shown,
yes,
if not all, are from clients or from applications that have been published. Yeah? The data are not ours. We do have data sets, and there's actually we have one data set, or a couple of data sets have been published from our from one of our students who also participated in some machine learning competitions, and they are public. Yeah? But there are a few data sets. We are currently also together with the Institute of Pathophysiology in Vienna in a project for machine learning recognition of some specific cell structures, and they also have public data sets. When you drop me an email, then I can bring you in contact with the person in TissueGnostics who can tell you exactly where you will find them. But these are specific data sets that we use for training or testing. From the data that I have shown, the data are not ours. Yeah? These are data that belong to customers, to clients, and they are published. Yeah? So I cannot, although I have many of the data because we collaborated with them, but they are not publicly available. They belong to our clients, and I would need to ask if they agree to share those data.
Thank you so much. Thank you again for the talk.
My pleasure. Thank you.
Okay. I can't see any other burning questions from anyone, but as Suzanne, have you got your hand up?
Hello. Hi, Rupert. How are you doing?
Hi, Susan. I'm very well, thank you.
Wonderful.
How are you?
Just wondered if you could maybe, I'm sorry, I missed the beginning of your talk, and I don't know if you've already said this, but what do you need to start working with your system? You need the software, obviously, but can you work with any microscope, or what do you need?
Thank you, Susan, for this question. I did not explain this. In fact, we offer complete systems, including the microscope hardware and camera hardware, but our software is also available as standalone version, and we support the most of the third-party slide scanners in the market, like Hamamatsu Nanosumer or the AxioScan Z1 from Carl Zeiss, Aperio 3D Histech. And what is also always possible as a kind of legacy mode is that you import generic TIFF, JPEG, bitmap, PNG files, or also big TIFF files. And with the latest version of our software, we are compatible with the Omero TIFF, also the OME, or Open Microscopy Environment standard for TIFF files. Yeah. So in that sense, you can use any slide scanner that you may have available. You can even use any microscope and manually pictures. You may not end up with a virtual slide, just with a bunch of individual fields of view, but still they can still be analyzed. So the software allows to import those images, import and analyze.
Great, thank you.
Thank you.
Lovely. Okay, are there any other questions from anybody who's still online? I think, oh, there's something in the chat. No, just to thank you very much. Yes, well, thank you very much, Rupert, and thank you very much everyone else for attending. As Rupert says, you know, there are lots of different ways to contact if you've got any other questions. I'm also happy to forward any questions through if you think of anything later on. So thank you very much. And
thank you for the kind invitation, and have a good day. Good afternoon.